Hi everyone, it's Dr. LeBlanc with Unit 5A Prokaryotic Gene Expression, the Lac Operon. In this video capture lecture, we're going to talk about regulation by the Lac repressor. Our learning goals are to learn how an operon is constructed, specifically the Lac operon, to learn what a polycistronic messenger RNA is and what benefits it has for gene regulation, to learn what enzyme adaptation is and how it is achieved by the Lac operon, and to understand how a transcriptional repressor and a small effector molecule function to control gene expression in response to an environmental stimuli. This is a diagram of the operator region that's in front of the Lac operon. Notice the DNA molecule is looped around, and the repressor protein, here shown in green, is bound at two different sites. Prokaryotic transcriptional regulation is based on cis elements of DNA bound by transacting factors or transcription factors, which are proteins. The phenomenon of enzyme adaptation was researched by three very famous scientists who eventually ended up winning the Nobel Prize for their experiments. They are Francois Jacob, Jacques Monod, and André Lwoff. They worked at the Pasteur Institute in Paris, and they focused on lactose metabolism in E. coli. And you can tell that they are definitely French because here they are in the lab with their lab coats on, and they are smoking cigarettes right there. Nice big ashtray. They're also doing a little light entertainment. The Lac operon is an operon. What is an operon? It is a regulatory unit consisting of a set of structural genes, genes that encode proteins, under the control of a single promoter, one promoter. An operon encodes a polycistronic messenger RNA. That means that you have one long messenger RNA with separate genes for different proteins. Each gene on that polycistronic messenger RNA has a start and a stop. This allows coordinated regulation of proteins that are involved in a common process. Here's a diagram of the Lac operon. It has regulatory regions up here, including the promoter and the operator, which we're going to focus on in this talk. It has a structural gene called the LAC-Z gene, another one, the LAC-Y gene, and finally the LAC-A gene. These genes encode beta-galactosidase, an enzyme that metabolizes lactose, a permease that it lets lactose inside the cell, and another enzyme called galactoside transacetylase that has a mysterious function that nobody really studies very much. If we look at this region of the Lac operon and enlarge it, we can see this controlling region for the Lac operon right here in a little bit more detail. It has a site for a catabolite activator protein, and we'll talk about that in a subsequent video lecture. It has the Lac promoter, and a promoter binds RNA polymerase, and then it has the operator site to which the Lac repressor is bound. Upstream from the Lac operon is another gene, a separate gene, called the LAC-I gene. The LAC-I gene has its own promoter, and it encodes the Lac repressor. The close proximity of the LAC-I gene to the Lac repressor is a coincidence. It does not have to be this close, as we will see in subsequent discussions of experiments that Jacob and Monod did. Prokaryotic transcriptional regulation, again, requires cis elements and transacting factors. So here's our transacting factor. This is the Lac repressor, and like I said before, it is bound to the Lac operator. Notice we already have a molecule of RNA polymerase bound at the Lac promoter. The steady state of the Lac operon is to have the Lac repressor bound. When there is no lactose present in the environment, which is normally the case for E. coli, the Lac repressor is produced from the Lac I gene and sits on the Lac operator, preventing RNA polymerase from traveling down the operon and producing a messenger RNA. What if an E. coli lands in that puddle of melted Dairy Queen outside the ice cream shop? Well, when lactose is present, inside the cell it's converted to allolactose, which binds to specific pockets on the Lac repressor protein. When this happens, a large conformational change, the repressor undergoes a large conformational change, and the DNA binding domains right here move and fall off the DNA. So in the presence of lactose, the Lac repressor falls off the operator, and the RNA polymerase molecule that has been sitting here can now travel down the entire operon and produce its polycistronic messenger RNA, which gets translated into beta-galactosidase, the lactose permease, and the galactoside transacetylase. The binding of the Lac repressor protein to the Lac operated DNA occurs by a helix-loop helix domain in the Lac repressor binding to the major groove of the DNA molecule at the operator. So inside the cell, this is what happens. When lactose gets inside the cell, it is converted initially to allolactose, and then the enzyme beta-galactosidase will cleave lactose into two smaller sugars, glucose and galactose. The cell can't use lactose the way it is. It needs to break it down further in order for it to enter the glycolysis and the TCA cycle. The steady state, the normal state for a bacteria, an E. coli bacteria cell, is to have a small amount of lactose permease present and a small amount of beta-galactosidase inside the cell. These two proteins are products of the Lac Operon. Okay, let's follow what happens when a bacteria cell then encounters a large amount of lactose. We're going to start here. When lactose becomes available, say the E. coli landed in that puddle of melted ice cream, the lactose comes into the cell through the existing permease molecules that are embedded in the cell wall and membrane of the E. coli cell. There's a small amount of beta-galactosidase as well inside the cell, which converts it to allolactose, which binds to the Lac repressor and results in the Lac repressor falling off the Lac operon. Once the repressor is removed from the operon, RNA polymerase can transcribe the operon, and the polycistronic messenger RNA will then produce many molecules of beta-galactosidase as well as permease. E. coli loves lactose, and so in the presence of lactose, it wants to be able to use as much of it as possible. So it makes lots of molecules of permease to let in all the lactose it possibly can, and it makes lots of molecules of beta-galactosidase to metabolize the lactose into usable sugars. When all the lactose has been used up, the Lac repressor now does not have any allolactose bound to these regulatory pockets on the repressor. So the repressor rebinds to the Lac operon, shuts down expression, and during the normal process of protein turnover, the pores for lactose to come into the cell as well as the enzymes to metabolize it are reduced to normal resting levels, where the Lac repressor is bound to the operon and only tiny amounts of the transacetylase, beta-galactosidase, and the permease are produced. This is a very interesting video simulation that you can access by this website. You can download it for yourself onto your own computer, and that's what I've done right here. If you click on these boxes, show legend, you'll get this, and show lactose media, you'll see this. This is what happens outside the cell when lactose is present. Let's look at the legend for a minute. So this is our DNA. This is the LAC-Y gene. This is the LAC-Z gene. It's going to produce beta-galactosidase, and it's going to produce the permease. This is lactose. We don't have any lactose here yet. And the messenger RNA is going to be indicated by a black line, and translation by this arrow. And here is RNA polymerase. So right now, the resting state with no lactose present, the Lac repressor, right here, is bound to the operator, and it prevents RNA polymerase from transcribing the gene after it's bound to the promoter. It won't progress down the operon. Let's add a little lactose. Right now it's not getting inside the cell because there's no permease present. Oops. This is a little bit of a leaky operon. So here's a molecule of RNA polymerase that's made it through the operon. It's going to produce some beta-galactosidase and some permease. So once the permease molecules lodge in the membrane, they will let some lactose in. Now we have some of this lactose coming inside the cell, and it's going to bind the Lac repressor and prevent the Lac repressor from binding to the operator. That will increase the rate of transcription for the Lac Operon, will produce more messenger RNAs, which will be translated into more molecules of beta-galactosidase and permease. So we'll let more and more lactose inside the cell, and it's being metabolized by beta-galactosidase into glucose and galactose, which is then further metabolized by the cell in the glycolysis and the TCA cycle. Eventually, all these molecules of lactose will be converted to the smaller sugars, and the Lac repressor will have no lactose that can bind to it anymore, and the Lac repressor will bind to the operator region and slow down the rate of transcription to normal levels for a cell that is not in the presence of any lactose. Okay, we're almost done metabolizing all the lactose. Here comes a repressor molecule. Now there's no more lactose in the cell to bind this, and RNA polymerase will not be able to transcribe the operon anymore. During the normal process of degradation, these molecules will be reduced in levels, as well as the permeases, and we return to our resting state. So in summary, an operon contains several structural genes under the control of a single regulatory region. Transcription of an operon results in a polycistronic messenger RNA that encodes more than one polypeptide. The Lac Operon has 3 structural genes that encode 3 proteins. The Z gene encodes beta-galactosidase, the Y gene encodes a permease, and the A gene a transacetylase. The Lac Operon also has an operator region that binds a repressor. The Lac-I gene encodes the repressor. Its location close to the Lac Operon is a coincidence. Cis elements are DNA sequences, and transacting factors are proteins that bind to the cis elements. Bacteria can adapt to their surroundings. In the presence of lactose, cells produce the enzymes necessary to metabolize it as an energy source. The Lac repressor is a transacting factor that binds to the operator of the upstream regulatory region of the Lac Operon. The normal state for the Lac repressor is to be bound to the operator. This state produces only very small amounts of beta-gal. In the presence of lactose, the small amount of beta-gal in the cell converts lactose to allolactose, which binds the repressor. The repressor undergoes a conformational change, falls off the operator. RNA polymerase then transcribes the operon at increased levels, producing more polycistronic messenger RNA for the 3 proteins. Beta-galactosidase cleaves the lactose into glucose and galactose, and permease lets more lactose into the cell. When all the lactose is converted, the repressor, minus the lactose, rebinds the operator, and transcription is blocked.